Welcome to the lecture series in food biotechnology. In this lecture, I'm going to discuss certain genetically modified crops and the application of genetic engineering in improving different aspects of crop plants. So, the benefits of GM technology in agriculture. GM stands for genetically modified crops or genetic manipulation technology. In food production, genetic engineering is at present used mainly in plants and microorganisms since these are particularly suitable due to their asexual reproduction and easy cultivation. The discovery of genome editing techniques have now made it possible to modify in a highly precise manner particular sequences of the genome, such as rewriting, amending, deactivating, or completely removing a DNA sequence. That is, when we know the genome of a plant, we can identify the genes which are crucial, which are important, which need to be modified, or we can change the alphabet of the gene itself by the gene editing techniques, thereby improving the crop plant concerned. Since the techniques of genome editing are applicable in principle to all organisms, this way the plants and the animals can also be modified. There are two main aspects which have to be considered when we are talking about editing plants. One is modifying or the input traits, and the other one is output traits. So when I say input traits, it means those characteristics of a plant which is relevant to its cultivation. So this modification of input traits is to increase the efficiency of the agricultural or the biotechnological production of certain human and animal foodstuffs or additives, or to enable their production in a given quality or quantity in the first place. That is, the plant already produces a component which you want, but by editing, you are either going to overexpress or slightly modify that product into something which is more useful for human consumption, or maybe to give more nutrition to the consumers. Or you are increasing, for example, for a plant which requires very high amount of water, thereby it cannot grow in whatever water scarcity is there. If you can slightly modify the input rate so that requirement of water becomes less, then it can be more widely cultivated. So like that, small editing things in the basic traits of how you cultivate the plant. And this can also be for making the plant resistant or tolerant to certain abiotic circumstances. A number of crops such as transgenic corn, for example BT corn like the MON 863 or the MON 810, MON standing for Monsanto, the company which produced that, and the GM, genetically modified potatoes, have been injected with bacterial genes to make them resistant to certain pests, while other plants like soybeans and canola were supplemented with genes to make them resistant to certain herbicides. BT cotton is a genetically modified organism or a genetically modified pest-resistant plant, cotton variety, which produces an insecticide to combat the bollworm. Strains of the bacterium Bacillus thuringiensis produce over 200 different BT toxins. Each is harmful to different insects. These BT toxins are insecticidal to the larvae of moths and butterflies, beetles, cotton bollworms, and flies, but they are harmless to other forms of life. The gene for coding for BT toxin has been inserted into cotton as a transgene, causing the cotton plant to produce this natural insecticide in its tissues. In many regions of the world, the main pests in commercial cotton are the lepidopteran larvae, which are killed by the BT protein in the genetically modified cotton they eat. This eliminates the needs to use large amounts of broad spectrum insecticides to kill the lepidopteran pests, some of which have actually developed or evolved pyrethroid resistance. See, usually we spray pyrethroid insecticides onto the plants to prevent the plants from being eaten by caterpillars. And remember, caterpillars are voracious feeders. The flies or the moths will lay eggs. The eggs will hatch to give the larvae or the caterpillar. The larvae will eat, and then go into the cocoon stage and will emerge out as the moth. But this voracious eating would actually reduce the yield of the plant and the plant very effectively. So when the larvae gets killed, the yield is indirectly affected. So the one option is to spray insecticides, which are basically pyrethroids in composition. Many of the larvae actually, or the moths, have actually gained resistance or have evolved resistance to the pyrethroid insecticides. So developing a GM cotton where the toxin actually is produced, so when the larvae eats it, it dies. That was actually a very big breakthrough in transgene field. So this spares the natural insect predators in farm ecology and further contributes to the non-insecticide pest management. Bt cotton was created through the addition of genes encoding the toxin crystals in the cry group of the endotoxin. When insects attack and eat the cotton plant, the cry toxins of the crystal protein are dissolved due to high pH level of the insect stomach. The dissolved and the activated cry molecules bind to the cadherin-like proteins on the cells comprising the bush border molecules, the brush border molecules. So the Bt actually, it is a protease that becomes activated once it reaches the gut of the larva. And the larva have an alkaline gut which activates the protease. So then this will go, the activated molecule will go and bind to the brush border epithelial cells. And so this will now separate out. So usually what will happen, the epithelial cells brush border will separate out the body cavity from the gut and allows for the access to the nutrients. So binding of this will disrupt that, and formation of the cry ion channels are formed on the brush border epithelia. So this ion channels and opens up the potassium ions. So the potassium ions will flow out from the cells to the outer side. So you will have an ionic imbalance which will result in cell lysis, as a result of which the caterpillar will die. So the death of such cells, the regulation of the potassium ions is lost and results in the death of the epithelial cells. The death of such cells creates gaps in the brush border membrane. So this is the bollworm which we are talking about. So what happens is that this is a Bt resistant plant. The crystal and spores are integrated these are ingested when the caterpillar feeds on the leaves. The crystal and the spores get ingested by the larvae. These toxins are now activated in the gut by the gut enzymes. So what will happen? The activated toxin will bind to the receptor. Subsequently, it will insert into the membrane, cause leakage of the ions. So this is the binding will actually activate the receptor. The ions will start flowing out. So the ionic membrane potential is lost, which results in the death, because the larvae will no longer be able to, when the brush border epithelial cells are lost, the larvae will slow down its eating. And finally, it will reach a condition where it is unable to eat and finally will die. Another example of the GM crops are the so-called glyphosate resilient Roundup Ready crops. This is also marketed by the agrochemical company Monsanto, which is also one of the biggest manufacturers of the glyphosate-based herbicides. Here, in the normal, so what happens is that this is your normal plant. When you spray with the glyphosate, this works on inhibition of an enzyme. We will come back to that later. So what happens here is that the normal plant will have an enzyme in the shikimic acid pathway which gets inhibited. The herbs also contain that. So when you spray it, when the shikimic acid pathway gets inhibited, the plant will die. But in this case, when we transform the plant by agrobacterium mediated transformation, the vector contains, or the TI plasmid will contain, a modified glyphosate resistant EPSPS gene. And this transgenic plant is growing up. You are spraying the plant with the herbicide. The plant's endogenous enzyme will become inactivated because of the spraying of the herbicide. But since the plant is transgenic, it has the glyphosate resistant enzyme supplied by the transgene. That is active. So the GM plant will survive, whereas the wheats or the other crops which do not contain the transgene will die because of the spraying of the herbicide. The second aspect is the modification of the output traits. Output traits describe the characteristics for which we are going to utilize the particular plant. Genetic engineering can be used to make individual natural products more processable, improve the content, or make them more digestible to humans. A typical example of this is the golden rice, which contains more iron and provitamin A than the conventional strains of rice. Another example is the potato grade Amphora, whose starch production has been improved. So this is the golden rice. The color of the grain itself indicates the presence of extra beta carotene. This is your normal rice. But there are regulatory issues which have certain rules regarding the cultivation as well as the marketing as well as the consumption of GM foods. European Union has actually approved the potato grade Amphora for starch production. Golden rice is a variety of rice which is produced through genetic engineering to biosynthesize beta carotene, a precursor of vitamin A, in the edible parts of the rice, which is the endosperm or the seed. It is intended to produce a fortified food to be grown and consumed in areas with a shortage of dietary vitamin A. Normally, the shortage of or deficiency of vitamin A every year is established to kill 670 million children under the age of 5. And in many cases, it can also cause irreversible childhood blindness. Golden rice differs from its parental strain by the addition of 3 beta-carotene genes or biosynthetic genes. The parental strain can naturally produce beta-carotene in its leaves, where it is involved in the photosynthesis. However, even though the beta-carotene is present in the leaves, it doesn't get produced in the endosperm. The search for golden rice or fortification started off as a venture in the Rockefeller Foundation in 1982. Peter Bramley discovered in the 90s that a single phytoene desaturase gene, this is called as the bacterial CRT1, can be used to produce lycopene from phytoene in GM tomato. So conversion of phytoene to lycopene can be done by phytoene desaturase. A single gene, instead of having to introduce multiple carotene desaturases, which are normally used by the higher plants. So normally higher plants have a series of enzymes, a series of carotene desaturases which are used. Here, instead of that, we are bypassing using only a single enzyme. This lycopene is then cyclized into its beta-carotene by the endogenous cyclase in golden rice. The scientific details of this rice were first published in Science in 2000, the product of an 8-year project by Potrikis of the Swiss Federal Institute of Technology and Peter Bayer of the University of Freiburg. At the time of publication, golden rice was considered a significant breakthrough in biotechnology, as the researchers had actually engineered a complete metabolic pathway, a totally new biosynthetic pathway into rice. A simplified overview of the carotenoid biosynthesis pathway is seen in the golden rice. The enzymes expressed in the endosperm of golden rice are shown in red there. They catalyze the biosynthesis of beta-carotene. From geranyl diphosphate, beta-carotene is then assumed to be converted to retinal and subsequently to retinol, which is vitamin A in the animal gut. So we have the geranyl phosphate, which gets converted by phytoene synthase into phytoene and 2-pyrophosphate. This gives, by the activity of CRT1 gene, this gets converted into zeta-carotene. Again, by the activity of the carotene desaturase, it gets converted into lycopene. This lycopene, with the help of lycopene cyclase, can be converted into alpha carotene and the beta carotene. This is the normal pathway. The first field trials of golden rice were conducted by Louisiana State University Agriculture Center in 2004, and additional trials were later conducted in Philippines and Taiwan and in Bangladesh in 2015. Field testing provides an accurate measurement of the nutritional value, and it also enables feeding tests to be conducted. Preliminary results from the field tests have shown that the field-grown golden rice plant produces 4 to 5 times more beta carotene than the golden rice which is grown under greenhouse conditions. In several countries, golden rice has been bred with local rice cultivars or crossbred with the American rice cultivar Coco Drive. So what happens is that once you have a golden rice plant, you breed it with the local rice cultivars and create a hybrid which can be cultivated locally. As of March 2016, golden rice has not been grown commercially, and its backcrossing is still going on in current varieties to reduce the yield drag. See, when you create a complete metabolic pathway, there is bound to be some repercussions. That might be in either the quality of the seed, its shelf life, its stored protein content or carbohydrate content, or maybe in the case yield, the amount of the yield per acre or hectare when you're talking about huge cultivation. So crossing this with the local cultivars and continuous back crossing can actually we used to evade some of these problems, and so far this has been going on, and we still have not had the commercial cultivation of golden rice. In 2005, a team of researchers in Syngenta produced what we call as a golden rice 2. They combined the phytoene synthase gene from maize with the CRT1 from the original golden rice. And they saw that the golden rice 2 produced 23 times more carotenoids than the original golden rice, which we are now calling as golden rice 1. That produced only 37 microgram per gram, and it preferably accumulated beta carotene up to 31 microgram per gram of the 37 microgram of the carotenoids totally. So this produced better yield. In 2018, Canada and the United States approved golden rice for cultivation, with Health Canada and the US Food and Drug Administration declaring it safe for consumption. This was followed by a 2016 decision where the US Food and Drug Administration ruled that the beta carotene content in golden rice did not provide sufficient amounts of vitamin A to make a nutritional claim. Health Canada declared that the golden rice would not affect allergies, and that the nutrient contents of the golden rice were the same as in common rice varieties, except for the intended high levels of provitamin A. See, when you design a new hybrid, especially if it is a food crop, something which you consume, you have to show that the product does not produce any allergy, or it is not toxic, or does not interact with the other metabolic pathways so as to be detrimental to the normal metabolic pathways. So those tests are usually done before sanctioning the commercial approval for either cultivation or the product to be marketed. In this case, Health Canada declared that golden rice would not affect allergy, would not cause allergy, and the nutritional composition was on par with the normal rice. The only difference was the extra high levels of provitamin A. And in 2019, this was approved for direct use as human food and animal feed, and also for processing in Philippines. So this golden rice was created by transforming rice with 2 beta-carotene biosynthesis genes, the PSI, the phytoene synthase gene from the daffodil, and the CRT1 gene, the phytoene desaturase gene from the soil bacterium Erwinia. The insertion of the LCY gene, that is the lycopene cyclase, it was thought to be needed, but later research showed that it already produced, that is the lycopene synthase cyclase is already produced in the wild rice endosperm. The PS5 and the CRT1 genes were transferred into rice nuclear genome. They were placed under the control of an endosperm-specific promoter, thereby this would be synthesized only in the endosperm, not in any other part of the plant. So they are expressed only in the endosperm. The exogenous IC5 gene has the most transcriptase sequence attached. So it is targeted to the plastid where the geranyl geranyl diphosphate is formed. And the bacterial CRT1 gene is an important inclusion to complete the pathway, since it can catalyze multiple steps in the synthesis of carotenoids up to lycopene, while these steps require more than one enzyme in plants. The end product of the engineered pathway is lycopene, but if the plant accumulated lycopene, the rice would be red, and we don't want the red rice. The second area for the application of genetic engineering in fruit production is the genetic testing procedure. This can be used in conventional breeding methods for diagnostic purposes and for monitoring the quality control in food. In cases where genes which are foreign to the species of plant is introduced, the outcomes we call them as transgenic animals or transgenic plants. If we are going to insert only species-specific genes, then we call it as a cisgenic modification. The next question is of environmental concern or the hazardous concern. What will happen, especially if you put a bacterial gene into a plant? What sort of a repercussion can we face? So, how to safeguard them? The food itself is actually a GMO or a part of one, because especially a typical example is the flower sour tomatoes. The food will contain microorganisms which have been genetically modified. So when you talk about dairy products such as yogurt and cheese, or beverages like beer or wine, or even baked foods or sausages, they can contain the microorganisms which can actually act as the starter culture. So the GMO will produce the food because your organism is a GMO, it will produce the food, parts of which are detectable in the end product. For example, when you can take a GM tomato and use it to create a tomato puree or a tomato juice, that remnant of that GM, whatever is the gene I'm looking for, would be present, traces of it would be present in the puree or the juice which I make. Some examples of this are the cornflakes from GM corn, ready meals with the transgenic soya, and ketchup from GM tomatoes. Flower sour, or otherwise we call them as the Zenica tomatoes. The food is made with additives such as sweeteners and flavor enhancers, aromas and secondary ingredients which are obtained from the aid of genetically modified organisms, usually bacteria and yeast, but which are not contained in the additive itself. So we have only very few testing procedures which can actually direct residues or detect residues of the GMO in the individual additives. The next major aspect of how food can be produced is by what you call as smart breeding. A typical example of this is the work done by researchers in the International Rice Research Institute, IRRI. They were able to add a genetic sequence which is naturally existent as a variation in only a few rice breeds, and this was then taken on or continued to the other rice breeds. As a consequence, the new rice breeds are better equipped to adapt to oxygen deficient environment, underwater, and are thus insensitive to floods. They can also stand in water for a longer time without drying, which is usually the case of prolonged droughts or prolonged floods. The next tool we are talking about is the CRISPR-Cas9 technique. This is the latest technique which has been used for genome editing, and this is being implemented quite often these days. So we have what you call as the molecular scissors. The DNA sequences can be made inactive using these molecular scissors. They can be cut out or complemented by a use of restriction enzymes. This method opens up the possibility, for example, of controlling the selection of existing DNA sequences in the modified organism. We will discuss more about these modified crops. There are a series of crops, plants which have been modified, for which approval have been given. And we have some food products also where these modifications have been applied. We will be talking about them in subsequent lectures. Thank you.